Great. Well, I'll just get started then. I assume if there are any technical difficulties, someone will start shouting at me. But you'll have to bear with me. You will. You'll have to bear with me. This is probably one of the most technical talks I've ever given, but probably one of the most comfortable doing it at home in my own office chair. So hello, my name is Charlotte Payne. I work at the Plant Endomembrane Group at Oxford Brookes University. And my particular research interest through my PhD and through the first year of my postdoc has been looking at how the plant endoplasmic reticulum forms its shape and the effects of changing that shape on other cellular structures and functions. So just as a bit of a background to the plant ER, the plant ER is composed of two morphologically distinct domains. There are the cisternae, which are relatively large flat surfaces. You can think of them as sort of lakes that are connected by these streams, which we call tubules, that are formed of highly curved thin strands of ER that join together at three-way junctions. And one of the most interesting and exciting aspects of the plant ER for me is that it's so pleomorphic. It varies massively according to cellular demand, the developmental stage of the plant, and across tissue levels. So if we just compare here, this is a standard image of the ER in a leaf epidermal cell in a 7-day-old Arabidopsis. And you can see here, you know, it's a relatively spaced out network with not too many of those big flat cisternae that we mentioned. But if we just move down slightly into the root tissue, we find that there is this massive shift in morphology towards a large number of cisternae and the whole network itself that's quite densely packed within the cells. And so through our research, we found that there's a lot of different ways that you can modify the structure of the ER. Some are abiotic and some I guess you'd call biotic. So biobiotic, I'm talking about drugs, and these drugs can include ones that you don't think of the ER as the primary thing they affect. So for example, the trunculin B. The trunculin B prevents the polymerization of the actin cytoskeleton, which runs underneath the ER. But by removing this actin cytoskeleton, we find that there's a massive shift in the structure of the ER. You get an increase in cisternae, a reduction in the number of tubules, and you also lose any directed movement in the ER. Whereas another drug, Brefeldin A or BFA, which inhibits protein transport from the endoplasmic reticulum to the Golgi complex, we find again a shift towards a lot of cisternae. But it becomes a much more dense network and has this kind of unusual patches of intensity through it, caused by the fusion of the Golgi membrane back onto the ER. And stress, funnily enough, does a similar thing in that it changes the shape of the ER. And so this is, for me, the easiest way to stress out a plant, other than trying to take it anywhere interesting, is to briefly heat shock plant tissue for 15 minutes at about 42 degrees. And you see it forms this super dense network with lots and lots of small cisternae and many tubules connecting it. But we can also modify the ER using ectopic protein expression. So if we take here, there's this control ER, which is highlighted just by transient expression of green fluorescent protein and red fluorescent protein targeted to the endoplasmic reticulum by an H-cell motif. You see that there's a relatively even proportion of cisternae and tubules. If we transiently overexpress some ER shaping proteins such as the reticulum proteins, in this case reticulin 1, we massively shift the arrangement of the ER in favor of a lot of tubules and relatively few cisternae. And this work was done by a lot of really, really great researchers, some of which in collaboration with our lab. But my baby, since joining the lab, has been trying to push the ER the other way, so trying to induce cisternae. And Verena Kriechbaumer and several others found this plant protein family, the Lunarpark protein family, and we find that in that case, transient overexpression of either member of the Lunarpark protein family causes the formation of lots of these large sheets. And so when I started working on the Lunarpark proteins and this ER structure and shaping thing, I really quickly realized that there was a big problem in how we talk about ER morphology in plants. And that problem is simply that our wild type ER is so highly variable. So if you take a look at the graph that's hopefully appeared on your screen, you can see a whole array of different ER structures ranging from very, very tubular to very cisternal. These were taken, these are a few cells apart in a tobacco leaf epidermal cell, infiltrated on the same day, not treated any differently to each other. But we have a huge range of different shapes and structures. And the problem is, when we treat it with drugs or we induce changes in the ER, what we're really doing is we're shifting the proportion of unusual looking cells. So we're not generating new cell structures, new ER structures within cells. What we're doing is we're just making it so that we see more of them. So this is particularly obvious on Reticulum 1 overexpression. If you compare Reticulum 1 at the top to the image of just the wild type ER below it, they look basically the same. But the difference will be when we overexpress Reticulum 1, we don't see any cisternae at all in any of our cells. So this becomes sort of the common problem with microscopy, where you see an image and you go, does it look funny? And the first response is, I need to see another 20 more to tell you if it looks funny. So before we started any work on ER structure and shaping, we realized that we needed to find a way of actually measuring ER structure. So in conjunction with Mark Fricker, who works down at Oxford University, we developed one of the first iterations of a program called Analyzer. Gotta love the acronyms, of course. And I know that this is a slightly scary user interface, but what we've decided to do is generate an interface that is adaptable for different imaging methods. So I know myself, I've applied it to light sheet imaging, confocal imaging, and I've even had a go at looking at some EM imaging with it. And it's very, very highly adaptable, but the actual process that you go through to analyze images and extract this information from them is relatively straightforward. So we start by loading in a simple basic image. We then do some automatic filtering and upsampling of the images. So this just increases the number of pixels in your images in a very conservative way, and removes some of that background noise that you get in all images. And then we use an opening function to identify cisternae, or these fat bits of the ER. So you can see them highlighted in the top middle image in magenta. So once we've found our cisternae, we can exclude them and we can go on to enhance the tubular network using phase congruency filters. And these are particularly interesting because they cope very well with changes in intensity, which you find you get along ER tubules. They're not consistently bright in one color. So once we've enhanced our tubular network, we can skeletonize it and run a single pixel wide skeleton along each identified tubule. And then we can take the negative image of our tubules plus our cisternae to give us the polygonal regions. So these are highlighted in pink down in the corner. And these are the areas of cytoplasm enclosed by our ER network and are sort of the negative image of it, and help us get like a full, complete picture of what's going on with our ER structure. And we published this software in 2019, along with a big multifactorial analysis of ER structure, comparing how different drugs and stress and protein overexpression affects the shape of the ER. And the most exciting thing about this to me is that we were able to analyze 184 time series relatively quickly and relatively easily. So it's the first sort of medium throughput program of its kind out there. And it's been really, really exciting to use. I should also add as a plug that if anyone is keen to apply this to their own work, do get in touch. We're also experimenting with trying it on some mammalian cells, which has been interesting. So back to ER structure and modifying it using a range of proteins. As I mentioned before, a lot of fantastic researchers have worked together to identify the reticulum protein family. This is a large protein family of 21 proteins plus some splice isoforms. And the ones that I'm particularly interested are sort of the earlier numbered ones, so 1, 2, 3, and 4. And these are proteins that are involved in ER shaping. So they form a W-shaped topology in the membrane, which is used to constrict the ER and induce membrane bending. And the membrane bending can be so intensive that it actually hyperconstricts the lumen of the ER to such an extent that you can't get anything running through the middle of it. So if we take here this image, in magenta we have Reticulum 1, and in green we have GFPHL, which is literally just the green fluorescent protein with an ER retention signal. And what you can see is that the luminal protein, which would normally be distributed across the entire ER, is held in these discrete puncture regions. And further analysis with Analyzer has shown that these peaks in GFPHL intensity of this lumen marker intensity correlate with troughs in RFP Reticulum 1 intensity, which shows you that these regions where the luminal marker is aggregating are regions where we have relatively little Reticulum 1 squeezing these tubules. We're still working in our lab on members of the reticulum protein family. We've been applying Analyzer to some of them, and hopefully we'll have some exciting publications on that in the future. So moving on to the Lunarpark protein families, the Lunarpark proteins are sort of my particular babies, and of the two of them, my extra favorite is Lunarpark 1, and that's only because Lunarpark 1 marks cisternae specifically. It's the first protein we found that's able to differentiate between cisternae and the rest of the ER network. These Lunarpark proteins are characterized by 2 transmembrane domains, a zinc finger and a Lunarpark motif. And so what we find is when we increase the expression of Lunarpark 1 or Lunarpark 2, we get an increased proportion of cisternae in the ER network. Conversely, we've been looking at some sulc lines with reduced or no transcription of Lunarpark 1 and 2, and we find that the loss of Lunarpark 1 and 2 transcription results in relatively fewer cisternae, and the cisternae are much smaller as part of a proportion of the total ER structure. So what this has indicated is that Lunarpark 1 and 2 play some sort of role in ER cisternae formation or maintenance. So I wanted to look at whether or not Lunarpark 1 might play a role in cisternae formation and how it might do that. So we thought that the most likely methods for Lunarpark 1 to induce cisternae is either these Lunarpark 1 punctae that you can form in the ER expand to form cisternae, and as you add more, the expansions just get bigger and bigger, or perhaps aggregation of Lunarpark punctae. They all merge together to form one big cisternae. So we tried to take a look at this on the confocals that we have at Oxford Brookes University. We have 2 very nice confocals there. But we found quickly that the samples would bleach, and this event was a relatively rare event. It doesn't happen regularly that we see a new cisternae formed. So instead, we went to Harwell to work with Alessia Candeo, who is an amazing light sheet microscopist. So on the right, you have some images of actual light sheet data of the ER of a tobacco leaf transiently expressing the Lunarpark 1 GFP, and you can see all these little puncta of Lunarpark moving around through the ER. And the most exciting thing is that light sheet imaging is very, very gentle. You can image a leaf for a very long period of time with limited bleaching and limited phototoxicity. So we're able to observe events over a really long period of time, which you just can't do with some of the harsher methods like confocal microscopy. And so what we found is we found a few instances of Lunarpark 1 forming cisternae through aggregation. So if we just look here, you can see all these little puncta coming together to form one larger cisternae. And then it remains consistent for an extended period of time. The same over here, where these aggregates all cluster together to make a single cisternae that then remains as whole for an extended period. So over about 24 hours of imaging, we only captured 36 events, but of those, we found that the majority of the formed cisternae remained stable. They remained together for a period of greater than 10 seconds. 18% collapsed within 5 to 10 seconds and 20% collapsed in less than 5 seconds. And the undetermined category is for 1 or 2 newly formed cisternae that then immediately moved out of frame where you can't see them anymore, which is an absolutely classic issue in any type of imaging. So while I've been at the lab and before I was at the lab and using the work of others, we've now developed this amazing molecular toolbox for modifying ER structure. So we can now make it form tubules, we can make it form cisternae, and we can also use drugs to affect it in numerous ways. So now we're starting to use it to look at some really cool, really interesting things. So one of the first things we're looking at is how changing ER structure affects the movement of proteins through the ER. So here on the left, we have in green photoactivatable calnexin. So what you can do is you can shine a 405 laser on this and it will turn on or photoconvert a lot of the green fluorescent protein, and you can watch it spread through a network which is marked in magenta with a normal HDL marker, just so I can see where I'm aiming the laser. And this particular cell has also been treated with the trunculin B to hold it still to make activation easier. And we're very interested into looking into how the particles move through this ER network. This approach has been attempted before, particularly with John Runyon's and Imogen Sparks, but we are trying to supplement it with some single particle tracking in the endoplasmic reticulum. So how can you see single particles in the endoplasmic reticulum? And the answer is very carefully. We can use variable angle epifluorescent microscopy to track the movement of particles. So normal epifluorescent microscopy, you have the sample and the laser beam comes straight down directly onto your sample. The other end, you have total internal reflection fluorescence, where you hit the sample at supercritical angles and you form an evanescent wave, which you can then use to look at just the first 400 nanometers-ish of a sample. Or you can use variable angle epifluorescence, which is where the angles are not necessarily supercritical angles, but you're still hitting your sample at an angle with the laser. And you can use this to highlight a very thin region just near the surface of the cell. So if we look here, we have an example of high expression of this photoactivatable GFP calnexin in the ER, and you can see there's a great resolution through time. Now, if you reduce this expression, you can actually make out the movement of individual particles through the ER. And by projecting where all these particles have been using a color-coded temporal projection, you can actually see the outline and shape of the ER. And so we have been looking into this data. We've been looking into how different proteins move through the ER. So these are examples of TAR2, which is a full-size protein, versus a modified calnexin, which is just a very small transmembrane domain. And working with Chris Tynan on the technical side with the microscope and Dan Rolfe on the mathematical modeling side out at Harwell, we're trying to get a handle on how changing the shape of the ER and how changing the molecules that we're monitoring affects how things are moving through the endoplasmic reticulum. So that's kind of a really brief overview of what we get up to. Do get in contact if you have any questions. It's kind of one of those, I'll just show you everything as a picture book and lots of stuff we can talk about. I need to say a big thank you to everybody at Oxford Brookes University and all plant endomembrane group members, past and present. the amazing help at the Central Laser Facility in Harwell, so Stan Botchway, Alessia Candeo, Dan Rolfe, Chris Tynan, and from University of Oxford, Mark Fricker. And one last quick plug, we do have a PhD studentship coming up. As some of you might have known, Chris Hawes passed away over a year ago now. And we have a memorial PhD studentship going in his name. The start date is January 2021. If you have anyone that's interested in protein production and transport at the plant ER nuclear envelope continuum, please get them in touch with us. We'd love to add some more people to our lovely growing lab. And just to finish off, I should say, like they do on all the Bulmers adverts, that all these images were taken pre-COVID, and we no longer sit so close together on the stairs for safety. So don't worry, we're behaving well. And thank you very much for listening.
That's cool. Thank you very much, Charlotte. That's some fantastic imaging, as we know from the Oxford Brookes team there. So again, I think you've blown people's minds. The questions are a little limited at the moment, but let me start with a couple of things. So first of all, trivial one. What does Lunarpark stand for? I've often wondered, actually.
Oh, so Lunarpark comes from this motif that you have it here, LNPKH. Okay. But in the mammalian literature where it was first discovered, it does a much better spelling of Lunarpark, and now we're stuck with it because we've taken it over to the plant side. And I think Lunarpark is also a theme park somewhere in America. And it is a bit of a roller coaster to work with, so you just keep that name.
Indeed. So a question about the phenotypes of the mutants you said you looked at. So you saw some molecular phenotypes there, but do those plants have any growth phenotypes or anything? Can you say anything about what the Lunarpark proteins are doing in the plant growth?
No, they seem to cope relatively well. There doesn't seem to be any extreme growth defects. I think part of that is the fact that they can still form a small number of cisternae as they grow. And we even have some knockout lines by Amy RNAi lines done by Emily Breeze. And I think we found that they grew pretty much normally. However, other groups have reported similar mutants that do have growth defects when you knock them both out. So we're still a little bit in the weeds on whether or not there should be growth defects.
It's funny you should mention Emily. So Emily gave a talk previously before the summer, and you can see her talk recorded as well on the Garnet site, but she's just asked a question as well. So, so are the phenotypes you showed, are they reversible upon the removal of the drug and heat stress, or are they like set in place then? How reversible are they?
So largely reversible. If you wash out BFA and LAT-B, definitely reversible. Heat shock surprisingly seems to linger for quite an extended period of time. I actually think I've imaged it for over an hour and it still hasn't gone back to normal. But I'd assume that it will eventually revert. It's just that then I immediately slap it in a slide and continue to stress it out. So maybe if I left it alone a bit longer, it would probably recover.
Right. Okay. So yeah, we got a question from Pavithran Narayana, and they asked, do you think ER structure change may also accompany changes in membrane lipid profile to allow for more plasticity of movement?
Yes. Yeah, we think we do. So two members of the Reticulum protein family are actually found to be involved in lipid regulation, and that's something that we're actively looking into. Yeah.
Okay, cool. Good question. Okay, so I think we're going to have to leave it there. Questions start coming in. I've got kind of a practical question about COVID. So you mentioned everything was taken there pre-COVID. So obviously you guys, you're doing a lot of microscopy, and that means a lot of concentration in small dark rooms and a lot of breathing and things. So have you got how are you managing that work with at Brookes? I guess you can't work at the same time as different people, or how is it working?
So we are actually back up and running pretty much as normal. So Chris Hawes, in his infinite wisdom, built all the microscopes rooms, had them built plenty large. So we can actually have, I think, up to 3 of us working on the confocals. I think most of the M's are up to date, although Joe McKenna would know a lot more about that than me. It's just using a lot more precautions, so things like face masks and face shields and making sure that we're very careful around one another. But luckily we have quite large rooms, so we're able to socially distance. Yeah.
Good.
If anyone wants to come,
yeah, I should
we're
say,
running.
I mean, if anyone's interested in collaborating with these guys, they've got a really fantastic facility, as I said, it's set up by Chris and John Runnians as well, and they're happy to collaborate with anyone who wants to use their expertise on this stuff. So